And I am really excited to introduce our speaker today, Nicole Wagner. She did her bachelor's, master's, and PhD at Trent University in Ontario, Canada. Her doctoral research focused on identifying physiological traits in zooplankton that are indicative of nutritional stress. After her PhD, she did postdocs at the University of Toronto, Cornell University, and Baylor University. And she is currently an assistant professor in the biological sciences department at Oakland University. And I'm really excited to have Nicole here today because she'll be talking a bit about cyanotoxins. And as folks in this room might know, I have been pushing to get folks here in Nebraska to think more about cyanotoxins. So I'm excited that you'll all be joining me in that opportunity for the next hour. So without further ado, Nicole, I will hand it over.
Awesome, thank you. So thanks everyone for showing up. Today has been great. I've had lots of great conversations with everyone who I've talked to, and I'm hoping to have some great questions and feedback on, mainly I'm gonna be talking about stuff I did during my postdoc because I am in year 3 of an assistant professor. I have data, it's just not very well analyzed yet. So we're gonna talk about stuff that I know will be interesting. And so yeah, I'm really interested in how sort of nutrient stoichiometry, so that's how Jess and I know each other from the stoichiometry world, and looking at how that affects growth and toxin quotas, toxin production in diverse cyanobacteria. We're going to talk a lot about lab cultures, but I'm hoping to show you that it's also happening in lake environments. So to jump on in, looking at some broad scale US data. So this is the 2017 National Lakes Assessment data that the EPA puts out every 5 years. They just did the 2021, 2022 one. It just became public. Unfortunately, I haven't analyzed the data, so this data set's a little bit old. But 2 toxins that they are measuring, microcystin on this graph in different colors for concentrations, and cylindrospermopsin on the other graph. And so the EPA goes out and they sample 1,200, 1,300 lakes every 5 years, and they measure a variety of nutrients. They're looking at organisms. They're looking at a bunch of stuff. And so this is their toxin data. And so out of the lakes here for microcystin, the yellow is low concentration and the red is high concentration. Blue is more the intermediate. And you can sort of see it's a diversity of lakes throughout the US, but looking at sort of the Great Plains area, there seems to be a problem. There's a lot of microcystins being measured. And cylindrospermopsin, another common toxin that cyanobacteria can produce. I didn't bother color coding it. This is generally fairly low concentrations that we're finding, so it's not as high as the microcystins. But again, sort of this Great Plain region, Texas, Oklahoma, seems to have the highest frequency of cylindrospermopsin that they analyzed. And generally, these concentrations were typically under 5 micrograms per liter. Whereas some of the microcystin ones were upwards of 100 micrograms per liter in really, really dense cyanobacteria blooms. And so we're looking at this, and being in Nebraska, we sort of know sort of what types of nutrients and stuff that are going into our waterways. So I wanted to take sort of a nutrient approach, and can we sort of map these then and look at the nutrients and start to see what we can think of making patterns. And so this is the data. Colors changed. I'm sorry, different presentations that I merged together. So now yellow is higher concentrations, where blue is lower concentrations. And this is the water quality data from these lakes. And so on the x-axis, we have total nitrogen on a log scale. So how much just nitrogen, it can be nitrate, it can be within cells, and how much phosphorus on the y scale. If we're looking at stoichiometry and beginning to think about sort of nutrient limitation, whether or not things could potentially be phosphorus or nitrogen limited, Guilford and Hecky came up with these really good relationships. These are by mass, so this the red line is N to P9. If things are above that, if you're finding data points above that, that means that this system potentially could be nitrogen limited. It's not a guarantee, but it's leaning more towards nitrogen limitation. And alternatively, if the data points fall below, the system could be potentially more phosphorus limited if it's above that N to P of 23 by mass. So when we're looking at sort of aggregating this data, the lower microcystin concentrations, we have an average NDP of 32 by mole or 14 by mass, with a median of 26, so falling right into the middle. And then when we're looking at concentrations that are higher than 1 microgram per liter, so more toxic, more biomass present, the average NDP is 37 or 17 by mass, and the median is 31 or 14 by mass. But both of them are falling in this range in between the N-limited and the P-limited. And we're calling these more of like potentially co-limited. It could be nitrogen, it could be phosphorus, it also could be light or something that we didn't measure. But there still is this pattern that as you're increasing the N to P, so from 32 on average to 37, you're getting potentially more toxin production produced at these higher N to P ratios, meaning that there's more nitrogen per unit phosphorus. And so we wanted to look at this, and so we went to the grossest lake we could find, which is Lake Fayetteville, which is located in northwest Arkansas, right by the University of Arkansas. It's this really tiny reservoir that is, basically there's a park around it, and it has almost a yearly cyanobacteria bloom. So this water is very bubbly, and you can almost see like the streaks of cyanobacteria on it. I filtered this. This was 60 mils filtered through that. So that's very, very green. Normally we can put like 900 mils through one of these filters, and that was 60 mils, and it was getting clogged. And yeah, you can see the dense sort of scums accumulating along the side. And so we wanted to sort of test this nitrogen to phosphorus ratio. Could we make toxins become more, could we make these blooms become more toxic if we add nitrogen? So this is what the data look like, just observational before we did any manipulation. And what you can see here on the one axis is that we have nitrate and nitrite, so sort of the dissolved nitrogen concentration that is in the lake through time. And that is in the gray colored circles. And so initially in March, April, before sort of the bloom really takes off, there's really high nitrate concentrations. This is mainly nitrate, there's basically no nitrite. Close to almost a milligram per liter. And basically, as soon as the water gets warm enough, these blooms start to really take off, and you see that nitrate more or less drop down to below detection limits. And then it more or less remains at detection limits for the remainder of the summer until the lake mixes and where you get a little blip after the lake mixes and more nitrogen is put up into the surface waters. The open circles are the microcystin concentrations on the other axis. And so what we see here is this big, huge peak right when nitrogen is really climbing down. You're getting this really big peak of microcystin happening. And so it sort of indicates that there could be sort of this nitrogen signal. Is it something like they're beginning to stress nitrogen stress and they're releasing this toxin or producing it at maximum amounts? Or is this just almost the right temperature? And so what we wanted to do is just sort of really examine this nitrogen hypothesis. And so we took water, we brought it back to the lab, and we added nitrogen, and we wanted to see how the toxin concentration changed. And so here's the biomass that we have. On the x-axis, we have the control in which we did nothing. We just took the water, put it in a bottle, left it for 5 days, and then measured how much biomass it was. And then we added 3 gradients of nitrogen, low, medium, and high. And that was 100 micrograms per liter, 500 micrograms per liter, and a milligram per liter. And this lake was nitrogen limited when we collected it. So adding the 500 micrograms per liter and the 1 milligram per liter, we got more biomass out as measured by carbon. Looking at the toxin concentrations, so this is microcystins, we begin to sort of see this is the total one, so adding up all of these congeners. So microcystin has many, many different types of congeners, and we're able to measure 6 or 7 of them. And these congeners are dedicated by their last part, so LA means leucine alanine, LR is leucine arginine, so they're just the amino acids in which they have in the toxins. And so looking at the total toxin concentrations, again, when we added lots of nitrogen, we were able to increase the toxin concentrations in these systems. And then breaking it down by the congeners, microcystin LA, again, sort of similar patterns. LR, similar patterns. RR and YR, we actually began to see a little bit of increase in the lower concentrations of nitrogen, not just only in the really highest concentration. But either way, it's sort of giving us an indication that as we add nitrogen to the system, we're able to increase that sort of toxin quota in which these organisms are beginning to produce. And so why could this be? Toxins have very different nitrogen concentrations. So this is microcystin LR, so it has the arginine and leucine side congeners, and I've circled all the nitrogens on here. There's a ton of nitrogens in this. The other common one that I mentioned at the beginning, cylindrospermopsin, again, there's a bunch of nitrogens in it. And the last common cyanotoxin that is produced within America mainly is anatoxin. And it's a relatively small molecule, but there is only one nitrogen. And so when we put these to sort of more stoichiometry, looking at their carbon and nitrogen quotas, microcystin has a CN of 5.7. The lower the number basically means the more nitrogen per carbon. Cylindrospermopsin actually has a slightly even higher nitrogen demand. Its CN is 3.5, or by weight, it's 16.8% nitrogen. And anatoxin is fairly nitrogen poor. It has a CN of 11.6. And so this gives us sort of an indication why nitrogen might be so important for this sort of toxin production, because these toxins have very different nitrogen concentrations. The other thing that's not insanely well known, although you think it would be, is just what is the carbon to nitrogen range of these microorganisms grown under different environments. And so I tried to gather as much data as I could. There's not a lot of data on this, actually. So most of the ones that have really high N values, these are experiments in which I have done. And otherwise, there's not a lot of CN data out there on these different phyla of cyanobacteria. But just to orient you, we have the genera here on the x-axis, and then their respective CN ratios as the bar. And so we can see that microcystis has a really wide range of carbon to nitrogen. It can go all the way below to around 5, so really, really nitrogen rich, almost up to 20, so really, really nitrogen poor. You're getting this really big plasticity. Where the other organisms, Dolichospermum, Aphanosomena, and Cylindrospermopsin, which is now actually called Raphidiopsis, welcome to the world of phytoplankton that is constantly changing, they're more homeostatic, as we say. So their C to N ratios is definitely not as plastic as the microcystis. These lines are the different toxins that we just talked about. So this dotted line is a stoichiometrist's favorite ratio, the Redfield ratio. For CN, that's around 6.6. And it's not exact, but you can say if it's below the Redfield ratio, it has really a high nitrogen demand. And if it's above the Redfield ratio, it would have not as high nitrogen demand, more of a high carbon demand. And basically what we see is the exact same thing as what I showed from the other graph, is that our N-rich toxins, our microcystin and cylindrospermopsin, fall below the Redfield ratio, and our anatoxin is above it. Additionally, I added on another toxin, saxitoxin, which actually has the highest nitrogen demand out of all the cyanotoxins produced. And just looking at the bars compared to the lines, we can sort of see that the toxins are almost always more N-rich than what the organism is. And so it sort of sets off this trade-off that's sort of building up this idea that we really need high nitrogen conditions to get these toxins to be produced, even though they're a tiny, tiny component of the overall N demand. And looking at why these ones, the Dolichospermum, Aphanosomena, and Cylindrospermopsin, why they might not have this huge range of plasticity is because they are, well, A, they're cool. But yeah, so they can fix atmospheric nitrogen. So they can take the 78% of our atmospheric nitrogen, they can break that triple bond, and they can use that for growth. And so that is likely the reason why they don't have as much plasticity, because they don't have as much nitrogen limitation, because they have basically an unlimited supply of nitrogen. And so what happens with these types of organisms is that they will always use like the nitrate, urea, whatever sort of nitrogen source there is first. And when they run out of that, then they create, for freshwater ones, they create these specialized cells that are called heterocysts. And these heterocysts, they basically lose the ability to make oxygen. So they separate nitrogen fixation from photosynthesis. And inside here, this is where all this nitrogen fixation takes place. It's super, super energetically expensive. It costs 16 ATP to break that triple bond and to use that into usable ammonium for growth. And so basically, nitrogen fixation is fixed there, and then it gets shuttled into the other cells called vegetative cells. And then the vegetative cells give that one carbon as a source of energy. And basically, within 24 hours of running out of nitrogen, these cells are more or less functional. So they're fixing nitrogen and trying to grow using the atmosphere. And if nitrogen is reintroduced to the system, basically they'll shut it off. They don't drop these heterocysts, depending on the species. So they will be there remaining on the filament, but their activity will be more or less shut off if new nitrogen is introduced, because it's too expensive for them to fix when there's freely available cheap nitrogen around. So this sets up a nice dichotomy. We have ones that can't fix, that have this huge plasticity, and ones that can fix that have not more homeostatic conditions. And let's try to think about how potentially nitrogen in the water source, not just atmosphere, is going to affect their toxin concentrations. And so that was a good chunk of my postdoc that we did. And so we grew a bunch of these lab conditions, lab cultures. This was one crazy experiment which involved 190 bottles or something like that. It was huge, where we're trying to figure out how nutrient addition, nitrogen to phosphorus ratios affect the growth and toxin quotas of this. So since we did this a bunch of times, we more or less did the same experiment over and over again with different organisms. And so we did the same experiment with an aphanosomena that is a nitrogen fixer. So it can access that atmospheric nitrogen and in theory not become nitrogen limited. We did this with a Dolichospermum, which I just told you could fix nitrogen, but this guy couldn't. So we can treat it more as a non-nitrogen fixer, and a Microcystis that could not fix nitrogen. And we grew them over a range of nitrogen to phosphorus. And you don't need to remember N to P. Basically, it's just a nitrogen gradient. We kept phosphorus the same, and we changed nitrogen. So we have low nitrogen conditions over here at the N to P1, and super high nitrogen conditions at the N to P of 100. Just think of it as a gradient of nitrogen. Today, we measured a bunch of stuff. I'm going to focus on the carbon and nitrogen, and then also the toxins that they were able to produce. So jumping right into the results, looking at the biomass, these graphs are going to look the same for a while. So on the x-axis, we have N to P. Again, think of like around the 0 as really, really nitrogen poor, and 100 as really, really nitrogen rich. And then on the y, we have just how much biomass was in these jars. And so our aphanosomena, that is a nitrogen fixer, more or less, I mean, there's more variability at the low end where there's very little nitrogen, but they more or less grew the same, regardless of how much nitrogen we gave them. This was not the case for the Dolichospermum that is supposed to be an N fixer, but was not. We see this sort of saturating function where under really low nitrogen conditions, we got barely any biomass, and then it sort of saturated out. And similarly, with the Microcystis in blue, we're seeing a similar pattern again, where we're having low concentrations, low biomass at really low N conditions, and high biomass at higher N conditions. Jumping over to the carbon to nitrogen ratio. So again, a low CN means more nitrogen, a high CN means less nitrogen, and across a nitrogen gradient. The nitrogen fixing aphanosomena wasn't all that variable. There was slightly elevated CN ratio, so slightly less nitrogen under the really N poor ones, but more or less around 6. The Dolichospermum, there was more of a hump shape, so it looked like it almost could fix nitrogen, and that nitrogen level was going back down, so there was more nitrogen in them, and then it would have like this hump shape, and then stabilize out slightly below 5. And the Microcystis, again, as we've seen, it's hugely plastic, so it was able to go up to almost a CN of 17, meaning that there's tons and tons of carbon compared to nitrogen. It's very, very nitrogen poor, but when you give it lots of nitrogen, it's able to produce a lot of nitrogen within its biomass. All right, coming to the good stuff, the toxins. So this aphanosomena produces cylindrospermopsin, that is an enriched toxin. Again, to remind you, the biomass is basically the same. The C to N is more or less the same, but yet we're seeing this sort of saturation curve of the cylindrospermopsin. So even though it's not really indicating nitrogen limitation, the toxins still are showing that there is some form of nitrogen limitation occurring, that we're getting a saturating function. With Dolichospermum, it produced a lot less toxins. This was an anatoxin that it produced, so it's a carbon-rich toxin, not a nitrogen-rich toxin. It produced very, very little. It's not zero, but it's very, very close to being zero on the really low nitrogen, and then slightly more under high nitrogen. And the microcystins from being produced by microcystis, again, sort of shows this saturating function. Now, we know that, well, other than the aphanosomena, we know that there was different biomasses. So let's try to standardize this so we can get at sort of more cell quota stuff. Looking at the cell quotas, even with the aphanosomena, you're sort of seeing the similar pattern, especially because that one more or less was the same biomass. So again, under really low N conditions, it's still producing less cylindrospermopsin, even though it has the same biomass and it has the same CN, more or less. Dolichospermum, when we cell correct it, it's more or less a linear relationship. Under low conditions, it's producing less anatoxin, and under high N conditions, it's producing more. Our prediction would actually be the opposite, because this is a really low N toxin. We would expect it to be producing more of this toxin under low N conditions than under high N conditions. And our microcystin sort of followed the same pattern. Under low N conditions, it produced less, and then under high N conditions, it sort of saturated out. So what is this telling us? There's sort of this common response that low N to P conditions for these cyanobacteria really decrease the amount of toxins that they were able to produce compared to high N to P conditions, or just if you're talking about a nitrogen range, low nitrogen produced less toxins than high nitrogen, regardless of whether or not it was a nitrogen fixer, and regardless of whether or not it was an N-rich or C-rich toxin. And so we wanted to look at sort of what are the other physiological variables that we could potentially go out in the field and sort of make correlations with these toxins. And so there is cyanophycin on this graph. This is a nitrogen storage molecule that cyanobacteria are able to produce. It's basically a peptide in which a bunch of amino acids are stuck together, and it's more or less just nitrogen that it can use if it runs out. And looking at the correlation with microcystins, it's basically a positive relationship. So if you have more ability to store nitrogen, you have more ability to produce more of this N-rich toxin. Cyanophycin is a pain to measure, so we wanted to see if there was something that was a little bit easier that would give us more of a better proxy. And the other indicator is phycobilin pigments. So this is a light harvesting molecule that cyanobacteria use in addition to chlorophyll, but it can also be a nitrogen reservoir. We'd sort of determined it's not specific just to light, it also has some nitrogen storage capacity. And there's a decent relationship between phycobilins and cyanophycin. So the more cyanophycin, the more phycobilins you have, so it's more ability to store nitrogen. So then, since phycobilins are pretty easy to measure compared to cyanophycin, we're like, okay, well, what's the correlations between the toxins and the phycobilins? And so on the first graph, we have our cylindrospermopsin data, in which we have the phycobilins on the x-axis and the particulate cylindrospermopsin on the y. And we had sort of these two populations, the ones that were fixing nitrogen and the ones that weren't. So we decided to analyze them slightly separately. And basically, we found positive relationships for both. More phycobilins, whether or not you're fixing nitrogen or not, leads to more particulate cylindrospermopsin. But the intercepts are different. The slopes are basically the same, but the intercepts are different. So the ones that weren't fixing nitrogen just had a basal level of cylindrospermopsin higher compared to the ones that were. And then looking at different growth phases is another interesting aspect, whether or not these cultures or these blooms are in more of their exponential phase or sort of more senescing in more of their stationary phase. Again, we're seeing similar relationships. So in the exponential phase, so these guys are growing as rapidly as possible. There's basically nothing limiting them at this stage. And we're still seeing a correlation between microcystin and phycobilins. So the ones that have slightly more phycobilins do have slightly more microcystins produced. And then when you wait to stationary phase, it becomes insanely amplified. So in stationary phase, the ones that have more phycobilins have a lot more microcystins in their cells compared to the ones that did not. So this is giving us a little bit of a hint that it seems like nitrogen storage, cyanophysin, and phycobilins may be able to be used as a slight proxy to look at how much toxin potential there is. And all of these experiments were more or less grown under really short-term conditions in which we change their nutrient concentrations, basically over a nitrogen gradient. We know in lakes that this isn't always the case. Some lakes are chronically low in phosphorus, some lakes are chronically low in nitrogen. And so we wanted to look at how does long-term adaptation, acclimation affect sort of this type of pattern. And so we did this. This experiment was actually set up before I even started. But basically for 4 years, we maintained cultures at different nitrogen supplies. So we started with our happy culture that is a dolichospermum that can fix nitrogen. And for 4 years, we changed its nitrogen supply. So we grew it under really low nitrogen, intermediate, and high nitrogen. Then after 4 years, we decided to do a common garden experiment and sort of see at how all the ion elemental composition changed. And so we were able to measure a bunch of stuff. This actually is not a toxin producer, so this is sort of a sidestep before we go. And apparently it's going on its own. That's okay, we're ready. Okay, yeah, so the ionome. So the ionome is an elemental composition extending past carbon, nitrogen, and phosphorus. So this is just the short-term experiment here. So we're talking about if we were to take that dolichospermum and grow it basically how we did the other ones, what would their elemental composition be? We only changed nitrogen in the media. We did not change anything else. This is basically just a nitrogen gradient. What we found was there was a decreasing relationship with the amount of boron, calcium, iron, and magnesium. So the nitrogen limited ones, the ones grown under really low end supply, had more of this element in their bodies compared to the ones that were grown in really high end supply. And alternatively, we're looking at the other ways, nitrogen was higher in the high nitrogen treatments compared to the low nitrogen treatments. So it's sort of what we would somewhat expect. This is a nitrogen fixing molecule, so there is that ability for them to access unlimited sources of nitrogen. But also potassium, right here, was also higher. So again, we only manipulated nitrogen. That is the only element, and this is just all of their different physiology differences. So now, looking at what happens when you grow them long-term. So that was the short-term results. This is sort of 2 of the long-term results. What we have here is an effect size and then all the different elements on the x-axis. And basically, if the error bar doesn't cross the 0, it is significantly different. They're 95% confidence intervals. And so these are the different adaptations. So when we grew them for 4 years under low, intermediate, and high N, and comparing it to the short-term experiment. And these guys were all grown under low nitrogen. So some of them were grown for 4 years under low nitrogen and then exposed to low nitrogen. Some of them were grown for 4 years under really high nitrogen and then exposed to low nitrogen. And what we found is calcium is really decreased in these ones that were long-term exposed to low nitrogen, and then we grew them under low nitrogen. Calcium was decreased. We also have magnesium decreased. We have higher nitrogen, and we have a lot lower phosphorus. So this is showing signs that their ionome, their elemental composition is basically evolving to these different nutrient concentrations, and that their basal, sort of what they need, is changing compared to short-term versus long-term. And then again, when we grew them under more intermediate nitrogen supply, again, you can sort of see changes really with the high N seem to be a lot lower than the other concentrations. So it was kind of a cool side project. Sorry, there's no toxins about that. Lastly, I do field stuff. I don't just hang out in a lab. So the other thing that we did was we went back to Fayetteville and we installed a water quality monitoring profile. And so here it is. So here's the dam. This is a secondary water treatment plant. Thank goodness they don't use it because it's really, really gross water. And here is our pontoon. And so basically it is a solar panel and it's more or less attached to a winch that has a data son on it, and it collects temperature, oxygen, pH, and conductivity throughout the water column. So it stops every half meter, takes a measurement, and then it goes back up, and it did that every 2 hours. So we got 12 profiles a day. And here's the resulting temperature contour plot. So if you're not used to seeing these, we limnologists like to switch our x-axis, so the surface is the top. So just think of it like you're sitting on a boat, sitting right here, and this would be the temperature as you go down to the bottom. And then we have time on the x-axis. And the color, the more red it is, the hotter it is, the more blue it is, the colder it is. And so this lake has a really stable thermal climb. So basically, the top is hot, the bottom is cold. And you can really see this. So in July and August, it almost got to close to 35 degrees Celsius, whereas the bottom was barely above 10 degrees Celsius. So it creates these great big gradients in different nutrients and oxygen too. Here's the oxygen profile. I switched it from red to blue. So blue is now good because there is oxygen. Red is bad. There's no oxygen in this lake. So when you're looking at it, there's a huge amount of area of this lake that is anoxic. There's just a good chunk of basically from around 2 meters below, there's no oxygen in this lake. All the fish are definitely hanging around the littoral zones because they can't swim down here and survive. Interestingly, though, we have these peaks in which we're seeing huge changes in oxygen. This graph is scaled really weird. This is 350% oxygen, so it's 3.5 times saturated above what it normally would be. This, I believe, was like 26 milligrams of dissolved oxygen in the water column, so it's very, very bloom. There's a ton of photosynthesis going on, and that was occurred here. What I want you to draw your attention to is these black arrows. Basically, every time there's this increase in oxygen, whether it's more yellowy or more blue, we almost see an oxygen intrusion going down through the water column. And it's like something happened in which oxygen is allowed to get into the water column, even though it's a stable thermal climb. So there's not a lot of water going on differences, but there's still some form of oxygen transfer. And that basically happened in June, again in July, in late June, and again in July. And so if there's oxygen getting down, and we know that there's a ton of nutrients down there, are these part of the nutrients that are fueling blooms? And when I mean there's a ton of nutrients down at the bottom, I mean there's a ton of nutrients. So remember, from 2 meters below, it's anoxic. And this graph is looking at the ammonium concentrations through time. We just sampled monthly, which is why it's really bumpy for this contour plot. But this red dot is 6,000 micrograms per liter, or micrograms per liter, or 6 milligrams per liter. I think most toxicity to organisms happens around like 5 milligrams per liter, so this is insanely toxic for a lot of life. There's also no oxygen, so not many things can live down there. And yeah, basically it builds up over the summer. This is phosphorus, looking at micrograms per liter. The red area is 300 micrograms per liter. And again, it's sort of building up. It does some weird stuff in August where it looks like it sort of decreased temporarily and then increased again. But again, so there's a bunch of nutrients in the bottom. And so we were wondering whether or not these nutrients from the bottom water could actually be fueling these blooms and potentially toxin production. And so that's what we did. We collected bottom water, this hypolimnetic, nutrient-rich water, and we added it to the surface in sort of a more controlled experiment. So we went out there, we collected the water, and then we added nutrients as we normally would, so nitrogen and phosphorus, or both of them together. And then we also added half the expected hypolimnetic flux that would occur within 7 days, the expected hypolimnetic flux, and then twice the expected hypolimnetic flux. It's really hard to get the hypolimnetic flux when you're dealing with like a massive lake, even though it's relatively small, and you shrink it down to a bottle. So that's why we had the concentration range, to see if we were potentially off, basically. And we measured a bunch of things, but all I'm going to show is the toxin concentration. And so here's the June sample. This is what the dissolved nitrogen was at the surface water. So basically it's non-detected here again. And when we added nutrients, here's our control and the microcystin to carbon. So it's sort of standardized to biomass. Here's our control, our half hypolimnetic oxygen flux, hypolimnetic oxygen flux 2 times, our inorganic nutrients, nitrogen and phosphorus, and then N and P. And really, we didn't see a whole lot going on here in June. And when we added inorganic nutrients, we actually got less toxins produced. So there was a community shift, likely from microcystis to more of like a green algae caused this community shift. In July, we began to see what was beginning to be a pattern. So again, there's basically no nitrogen in the surface water. When we added nitrogen, all of a sudden we're getting more toxin production. So there's more toxins produced per carbon. And then when we're beginning to look at the hypolimnetic fluxes, again, it's not significant yet, but we're beginning to sort of see this trend as you're adding more of that bottom nutrient-rich water, we're beginning to see toxins increase. August, we finally seen it happen. So at twice the expected hypolimnetic flux, we were able to more or less get the same amount of toxins produced as our inorganic nitrogen. And again, when we add phosphorus and N to P, we see a decrease likely caused by a community shift from microcystis to other things that aren't producing toxins. And then in September, we lost the pattern. The lake, it was beginning to have a little bit more nitrogen. The lake was beginning to mix for the fall season. And we more or less, our hypolimnetic fluxes didn't more or less do anything. And there was a lot more variation just within the amount of microcystin and carbon. And so what this is telling us here is basically we have the biomass that's more or less consistent between the controls, the hypolimnetic fluxes, and even the nutrient additions. And so really what we're seeing is just sort of the toxin changing because the biomass was more or less the same. And so once the lake becomes nitrogen limited in July, adding that hypolimnetic water, so adding how that oxygen got down, if it brought up nutrients with it, it potentially could be fueling these blooms and causing them to become more toxic. And so this is why you're seeing this increase in microcystin concentrations in this lake. And yeah, adding the inorganic nutrients can really change with the phytoplankton community. So when we're adding a lot of phosphorus to this, it seems like greens more or less took over from the cyanobacteria. They were able to grow faster and just outcompete the cyanobacteria. So our toxin concentrations went down. But that didn't happen with the hypolimnetic flux. So that N and P in the hypolimnion that you could use to potentially fuel these blooms, it seems like microcystis really likes that and was able to then produce more toxins. And so really what's the big story here is that we need to be able to control the nutrients coming into our lake. We've done a great job, or a semi-great job, at controlling the amount of phosphorus into our lakes from the 1970s up until now, but we basically ignore nitrogen. And so by ignoring nitrogen, we can really throw off this N to P balance, and creating excess nitrogen, we could making potentially these blooms worse or shifting the communities to want to have more cyanobacteria compared to other types of phytoplankton. And so, yeah, I'm now team co-limitation, that we need to control phosphorus loading and nitrogen loading to really get these blooms under control. And I'm gonna guess I talk fast, but that is all I have. So yes, I want to thank everyone that helped. Science doesn't happen in a vacuum. This was really done with our Oceans and Human Health Center that was based out of South Carolina, and most of the work was done during my postdoc at Baylor. And have tons of time for questions, so please bring them on.
Nicole, any questions?
So this was applied to one lake in Arkansas, correct?
It was, yeah.
Could this also be applied to other lakes across the country?
Yeah. So actually, now that I am in Michigan, I'm beginning to do sort of very similar stuff in there, and we're finding similar results. I think Nebraska is a great example for this. There is a ton of nitrogen in these lakes, and I think if it does develop a stable thermocline, which might not happen here as often as other places, there could potentially be this internal nutrient flux that might help be fueling blooms. Yeah.
And my other question is, would you see similar results sort of in the areas where sewage may leak out into oceans or sort of estuarine systems near the ocean, or since there's more of an oxygen flow in those systems?
So yeah, actually at the base of the Gulf of Mississippi, I mean, there's a hypoxic zone there that doesn't really produce toxic cyanos. That's just more of an algal bloom.
Mhm.
But around the Gulf of Florida, red tides are really common, and there definitely has a nitrogen signature there. Parasitic shellfish poisoning is basically saxitoxin, so it's hugely nitrogen costly. And yeah, like more or less where they're coming, where those blooms occur are where there's a bunch of rivers and stuff that enter into the Gulf. So I'm guessing that they're getting a bunch of nitrogen from there.
All right, thank you.
Thanks, that was really cool. I've got a question about the first set of experiments you were showing, where you get that shift in toxin production and kind of levels off. Does that correspond with like a shift to phosphorus limitation? Because you were showing some graphs where in the exponential growth phase, right, they're not producing as much toxins as the stationary growth phase. So are they just shifted to phosphorus limitation? They don't know what to do with all the nitrogen. They're just putting it into toxin production.
I would like to say yes. The answer was we didn't actually grow these long enough to actually get phosphorus limitations. So while the N to P was 100, there still was basically SRP measurable in there. We needed to grow them longer, but basically I think they were becoming light limited at that point. So we need to lower the phosphorus concentrations to actually see NNP limitation. We had it at a relatively high phosphorus concentration just so we could get the biomass. And I think we overestimated. We should have lowered it a little bit to have N and P before light limitation started to take off.
Okay, but so still that relationship between growth, you don't get toxin production versus once you start to get limited by something you do.
Yeah. With phosphorus limitation, I have done one study, I didn't show it. It actually increases slightly a little bit more of the toxin because there's a lot of excess nitrogen around. So when they do shift between N and P limitation, you actually see a slight increase in toxin production just because they have a bunch of nitrogen and not phosphorus.
Cool, thanks.
Yeah, thank you for that. Yeah, I have a question for the resupply of oxygen in the reservoir. Do you know what might have caused that?
It's a good question. Do you know? So this was really close to the dam. So I wonder if it was some form of like internal wave that was potentially coming down or something like that. I really have no idea. That I get asked that question every time. You would think my answer would improve after 2 to 3 times. We don't know. I'm actually really interested to see if it happens in other reservoirs or is this just a random Lake Fayetteville thing that we see?
Yeah, it's interesting to know. Maybe look at the hydrodynamics and all.
Yeah. I mean, the thermocline is super stable, so I wasn't expecting there to be oxygen there. I don't understand how it got there. If somebody else does, please enlighten me.
Oh, my question. But following on that, and I don't remember how the temporal aspect of that oxygen push, was it fairly quick? Was it like a 24-hour, 48-hour period or something?
They varied between like 24 to probably 72, maybe upwards of like 96 hours. So it wasn't a very long period. We're talking a matter of days.
That, but it was measured, it would have been measurable if, you were out there on a fairly regular basis. It's not just continuous monitoring, I guess, is.
That's a good question. I mean, it really would depend on the day that you go out there. Like if you're going on a weekly sampling, you might never see it. And you would only observe it with this high frequency monitoring. Or there could be times where you might see it and then be confused why there's oxygen in the bottom where the week before there wasn't and the week after there wasn't. So
Yeah, because the fisheries folks here are taking temp DO profiles, but it's whatever day they're out there. So it's kind of an interesting thing to think about, that maybe they should be looking longer term.
yeah, I mean, these buoys are coming down in price, so, but they're a pain to maintain. So I don't recommend it unless you have really good students and postdocs that love going out all the time.
So I'm curious on your how we got to your last statement. Right? We've paid attention to phosphorus. We say, oh, that's really going to tropify lakes. And there's all this evidence you presented that nitrogen is causing all these problems. So were we not paying attention to the microcystin? How did we get to this kind of canonical idea that phosphorus is what we need to pay attention to, and it's both?
Yeah, so initially in the 1970s, basically lakes were turning green and we didn't know why. And there was an interesting sort of bottle bioassay stuff that I do versus the whole lake experiments, in which they're looking at whether or not what is truly controlling production. And in the 1970s, they did this experiment at the experimental lakes area in northwest Ontario, so really far up north. And basically, they found that phosphorus was more or less the limiting nutrient. And so if you add phosphorus, you're going to get an algal bloom. And if you don't add phosphorus and you add nitrogen and carbon, you don't really see that algal bloom because phosphorus is controlling the production. And so then this set off, this was a good chunk of the water quality agreement between Canada and America to really try to reduce the phosphorus inputs into our lakes. And so Lake Erie is a great one. In the 70s, it more or less was classified as a dead lake. We decreased the amount of phosphorus. It came back, it revitalized. Dr. Zeus even changed the story in whatever that book was. It no longer went to Lake Erie for the dead lake. And then again, starting in the 2000s, all of a sudden we start to see these blooms again. And so we did do a good job at getting rid of phosphates from our laundry detergent and all of the other aspects of it. And it really did clean up a good chunk of our lakes. Basically, more or less, as soon as people said phosphorus is the problem, there was people out there being like, I think we should also be concerned about nitrogen. Those people just didn't get a strong enough voice. And so they more or less lost out, partially also because controlling phosphorus is decently cheap. To remove it from water treatment plants, controlling nitrogen from water treatment plants is really, really expensive. So the policymakers were like, yeah, we're just going to do phosphorus. And I think now there's more evidence that really we should be concerned with both, not just controlling P, because we're really shifting the imbalances of these communities.
I have so much to say, but also I feel like in Nebraska, we talk about nitrogen really well and not
Good.
phosphorus.
Good.
But I'm like, both. We need to talk about both.
We need to talk about
But one of the things that I was thinking about is, following along with this, the generation of the phosphorus paradigm. But there was Val Smith's work in the 80s where you saw that just cut off. You just did not find cyanobacteria once you got to a high enough N to P ratio. And I'm kind of wondering your thoughts on this, the balance of talking about total nutrients in the system versus relative nutrients in the system, and how that maybe is playing into our understanding of cyanotoxin development. And I'll back up a little bit and say, too, I would say we don't have a good understanding not only of cyanotoxin production, but the intracellular versus extracellular amounts. And
Okay.
Anyway, there's a question in there somewhere. And
Yes. Okay. So yeah, The total versus relative, so like talking about dissolved, sort of like what's bioavailable, I'm assuming that's what you're talking about?
Yeah, well, just the fact that like, at a certain, is there a certain point where it doesn't matter what the NDP ratio is of a lake if there's just chock full of nutrients? I mean, I would argue that.
Yeah, so I mean, I think, so like when Val was finding that basically cyanobacteria are not found in these really, really high NDP lakes, those were mainly oligotrophic. So there was very little phosphorus in there to begin with. And so I think to get a cyanobacteria bloom, you need a certain level of phosphorus. It needs to be semi-eutrophic. And then that nitrogen really can control the amount of toxins that's being produced. But yeah, if you go to like, you know, good places in northern Ontario that's on like the shield, basically there's no phosphorus and there's a bunch of nitrogen from nitrogen deposition. And so the N to P ratio is like 100 to 200, and you would think that that would be a perfect spot for like blooms to happen from what I just told you. But because there's so little phosphorus, there's basically just very little cyanobacteria. They can't produce enough biomass. So I think you need the biomass component, so like phosphorus really controls the biomass. If you have no phosphorus, you can't have a lot of phytoplankton. You increase the phosphorus, you can have this diversity of phytoplankton. And we know as you increase the phosphorus, we do get to see sort of the likelihood that the community is going to be dominated by cyanos. And so at that point, then it's sort of, is it N-fixing cyanos or non-N-fixing cyanos? And so the non-N-fixing cyanos are more on the higher N to P, and the N-fixing cyanos show up more on the lower N to P. And so I think that's where a lot of this stuff lies. And most, shouldn't say most, a good chunk of eutrophic, so high nutrient lakes, they have a ton of biomass. So there's a ton of just other organic processing happening. So they tend to have really high denitrification rates, which causes their N to P to be on the lower side than like an oligotrophic lake. So I do think it is more of this more relative, like, because yeah, we see lakes that have a decently low N to P. Lake Erie, for example, in August, it oftentimes has a really low N to P, but is producing a ton of toxins. So I think it does have sort of more to do with like the relative amount and how fast it's cycling through the community that's there.
And then a quick question on your earlier experiments. Were you looking at intracellular cyanotoxin, extra, or both?
Both. Okay, we had both. Interestingly, microcystin is more or less kept inside the cell unless it dies, and then it becomes extracellular. It's very low concentrations in the environment extracellularly, unless there's like a virus or something that is causing a bunch of cell lysis to happen. Anatoxin can be found in decent concentrations in and outside, and our cylindrospermopsin, at really high N concentrations, it seemed to be pushing a lot of it extracellularly. So it was both internal and external.
Yeah, so I have a question about the influence of the increase of cyanotoxins in the lakes to the other organisms. Like, I know it can harm a lot of like animals, like fish, but how about to the bacteria? So when cyanotoxins concentrations increased because of an concentration change in the environment, are there some bacteria like can do degradation of cyanotoxins? How will that like influence the concentrations of cyanotoxins?
So there is a couple different types of heterotrophic bacteria that can degrade microcystins, and they're pretty efficient at it. So yeah, there definitely is. The community of heterotrophs can actually influence the toxin production. I don't know whether or not it's like allelopathic chemicals switching back and forth or what it is, but there's been some cool studies out with like swapping heterotrophic bacteria from like a non-toxic bloom and putting them into like a toxic culture, and then taking one from a toxic bloom and putting it into a toxic culture. And the heterotrophs isolated from like the toxic culture can seem to make microcystis produce more microcystin than the ones that were isolated from the non-toxic blooms. So there's definitely a ton of communication that we know absolutely nothing about, and I am super excited to dive into that area. It's definitely a future area for me, but currently we don't know much. Regarding like fish and other things, it doesn't overly affect a lot of organisms. I mean, it definitely does. Fish definitely do get microcystin. There has been studies out there that say Daphnia are really sensitive, but then there's equally a number of studies that say Daphnia is not sensitive. So I don't know if these are like Daphnia clone differences or what, but I don't think there's really a consensus out there yet on exactly how this toxin affects all the other aquatic life.
Thank you.
Anyone else? Okay, so I think this is it for today. Thanks everyone for coming here.